Hello, everybody. How's it going? Thanks for joining us again for another live presentation. Today is a particularly exciting live stream all about shark bacteria, something that I didn't know could be so interesting. We're really glad that all of you are here, especially because, at OSearch, there's a lot going on. You see us putting the tags on the sharks and tracking sharks in real time, and all that's really, really cool. But we're also supporting up to 18 different research projects. And so it's all going way beyond the tags and the tracking of the sharks. And today is an excellent example. So we're really glad to have so many of you here to learn all about shark bacteria. Today's presentation is presented by BACD. They're one of our partners. They actually do have some supplies back in stock. So if you do visit bacd.com and you use the promo code OSearch at checkout, you can get free shipping and make sure that a portion of the proceeds go to funding our research. So thank you. I am joined here with some awesome people who are actually going to be leading the conversation today on shark bacteria. First is Dr. Kim Ritchie from the University of South Carolina at Beaufort. She was just recently our chief scientist on the NASA expedition. Kim, you've been working with OSearch since 2015 or so?
Yeah, yep.
Excellent. And her study is specifically on bacteria, beneficial bacteria, and so she is really the expert in all things bacteria. We also have Jen Cotton, who is one of OSearch's education ambassadors. She's been a teacher in Florida for about 10 years, right, Jen?
Yep.
She's been instrumental in helping us build up our STEM curriculum. So huge thank you to these two for joining us. Jen has also been on board for several expeditions, helping lead our education outreach. And so huge thank you to both of you. One quick question before I get started and I turn the conversation over to them. If you guys do have questions, please, please, please leave them in the comment section and we will get to them either. Generally, I'll try to save all the questions until the end, but if there is a really good one that I think needs clarification right away, I will interrupt and ask that. Anything that we don't get to, I will make an effort to go back through all the comments and answer them for you. So with that, I will turn it over to you, Jen, to introduce a little bit about today's topic.
Great. Hi, everyone. My name is Jen, and I wanted to thank you for joining us today and being part of the OSearch community. Our OSearch education programs and lessons are all available for download at osearch.org. I have linked all the resources for you, and you can also email us at any time at education@osearch.org, and we will definitely be getting back to you with that if you have any questions. So today we're going to look at an OSearch collaborative research to see how we're advancing in bacteria studies. So Dr. Ritchie, can you please tell us a little bit about marine host microbe mutualism?
Yeah, thank you. Well, I've been working on marine microbes since the early 90s, and I started out working on coral microbes.
Will you please mute your mic?
One thing I wanted to reiterate today, if anybody else is in a pandemic spiral every now and then, the big lesson from today is that the majority of microbes on the planet are good. They're good for the planet, and they're associated with every single organism on the planet. And the next slide kind of illustrates that corals, which I've worked on for many years, have specific bacteria associated with them. I started working on sharks just a few years ago when I was at Moat Marine Laboratory and started looking at bacteria associated with stingrays with my wonderful collaborators Carl Luer and Kathy Walsh. And we found that there are an awful lot of bacteria associated with stingrays and corals and other organisms that might help protect them, that produce antibiotics for them. So that's kind of the benefit that I'm looking at there. And so one thing that we've been doing with OSearch is specifically looking at bacteria on white sharks. And there are some things I wanted to reiterate about humans too. So humans also have beneficial bacteria, and this is probably an association that's been studied more than other associations. So we have bacteria in us and on us that do good things for us. We have bacteria that outcompete pathogens. We have bacteria in our gut. E. coli actually makes vitamins for us. You can't make vitamin K on your own. So they play these beneficial roles. Let's see, the next slide looks like here. Okay, I'm just gonna highlight some research, I think.
Sharks and rays have this amazing ability to heal their wounds very quickly. My part is to see if there's a beneficial microbial component, and from what we've seen in stingray research, there is. There are a high percentage of bacteria that may act as an immune system, that first line of defense for sharks and rays that produce antibiotics. We're running out of antibiotics that work worldwide, and this is a novel source of new antibiotics and another avenue of interest for some lines of research.
So Kim, that's really interesting. How do you go about determining whether a bacteria might be beneficial to the shark?
Okay, well, I'm interested in a couple of things. So are these bacteria actually beneficial? So what we do is culture them. So I will swab them from the surface of the shark and various different surfaces. And one thing that's kind of cool that you can see in this image that seems to be true with white sharks that is not true with some of the other sharks we've looked at is that they come in all different colors. They have these really beautiful colors. So this little bit of data that I'm about to show you is from a couple of white sharks that were tagged by OSearch off of the coast of Hilton Head. One was named Hilton and one was named Savannah.
So, Kim, I'm really curious here. How do you actually, can you go into a little bit more detail? How do you actually collect bacteria from a shark?
All right. Well, in that video that you just showed, it showed us swabbing them. So we kind of take sterile swabs and just swab them, and then we rub the swabs on those petri dishes that you see in the figure up there. And then they grow. They just grow and divide for a few days until we can subculture them to purification. And what you're looking at up there are purified bacterial strains, those different colors of dots on the plates, little agar plates. And that's how we do it. We have to get them in pure culture first, and then we can start to assay them.
So what parts of the shark specifically are you actually swabbing?
I'm swabbing every surface I can reach, essentially. So dorsal, ventral, teeth, eyes, gums, nostrils, gills, cloaca, any type of surface to see if there's a trend in the number of bacteria that produce antibiotics, or if there's a trend in the type of bacteria that I find on the shark.
So
is this something that's scary? Like a lot of people think like, oh man, you're on the deck with this big white shark and you're going down and you're swabbing it with these little Q-tips. I think to a lot of people that's a little bit unnerving. What is that experience kind of like?
Well, you think it might be, but then Brett and the crew have such good control over the animals and they're taking such good care of it that all you're worrying about the animal. All you're thinking about is to get your swab so you can get off so they can get the shark back in the water. So it's different than I expected, but not scary.
Jen, you had a question?
Yeah, so antibiotic resistant bacteria is kind of on the rise and a big concern. So can these antibiotics be used to help kind of combat that issue that we're having?
Well, that's the other avenue. So I'm interested in beneficial microbe associations with their hosts, like what they might be doing. So that area of research is pretty much mutualism, like mutually beneficial organisms. But another avenue is just what you mentioned. There's a lack of antibiotics that are working on certain bacteria that are becoming resistant to our current antibiotics. So this is a novel source of antibiotics that nobody's really looked at. So we have this library of thousands of different bacteria from these sharks that can be screened for antibiotic activity. So I'm looking for a benefit to the host by looking for antibiotic activity anyway. So when we find it, they could potentially be used to create new antibiotics. And you're kind of looking at this bottom panel here on the bottom right. I think this might be the only panel where I show actually antibiotic activity, that little halo or that little zone of growth inhibition around little dark spots are spots where antibiotics and microbes bacteria produce antibiotics. And they do this as a chemical weapon. They do it to secure a niche. They don't have fangs or claws, so they produce chemicals to talk with and to defend themselves with. So that's kind of what we're harnessing for hopefully medical purposes.
So real quick, Kim, here, obviously you're swabbing all over sharks, which means that they're basically covered in bacteria, right? And I think in a second you're going to show us what some of the bacteria is that you have found and where it is, and go into a little bit more detail on that. But I did get a good question here, since we now know that sharks are actually covered in bacteria. Can sharks get sick from this bacteria that's covering them?
Well, I think anyone can get sick from bacteria, even if they're beneficial, if they find themselves in a part of the body where they're not supposed to be. Just like when you think about E. coli, if it gets in your urethra, you can have a urinary tract infection, even though they're good for you and your gut. But sharks don't get infected as often as you might think, and I would defer to the veterinarians on that, but I'm certain that they probably get infected secondarily, just like we do, especially we have beneficial bacteria on our skin, Pseudomonas species, and these can become opportunity problem if you have burns. And I would imagine the same would be true with all organisms.
Okay, thank you.
All right, so this is really just showing how we make the culture library here. We end up cryopreserving them, so we actually freeze them down. So as I mentioned, we have thousands of these bacteria purified and frozen down. And what you're looking at here on the right side are rectangular agar plates that are our assay plates. So we end up letting these bacteria grow and produce their antibiotics. And after a few days, they produce these secondary compounds that kind of ooze out into the agar surrounding them. And then we overlay them with various different pathogenic tester strains like MRSA, which is methicillin resistant Staphylococcus aureus, vancomycin resistant Enterococcus, and various other gram-positive and gram-negative test strains, including human pathogens and marine pathogens, because I'm interested in what these bacteria might be doing for the shark. So I want to look at marine bacteria, but also can they be potentially used in drug discovery? So we look at human pathogens as well.
So real quick, Kim, can you walk me through, we see these swabs in the lab here. Tell me about how you go from on the platform with the shark, with the swabs, to immediately what happens after. So walk me through like a typical experience with you when a shark comes on to the platform until you get to where you are right here with these swabs that are growing in the lab.
Okay, so I or one of my students or postdocs will be one of the first ones on the lift because I want to get my samples and get out of everybody's way. So I get on, and usually Brett, who's the main fisherman, will help me with the head region. So we have to get samples of the teeth, the eyes, all the different regions. So I start at the head and work towards the the tail end. And as soon as I'm done with my samples, I get off and everybody's still working on the lift, and I immediately start plating them onto these round agar dishes. And you can see some examples here on the left side of the screen. Literally just swab them, spread them out, try to get them all spread out, and then let them grow for a few days. And then we can start subculturing them. So you literally just pick colonies. So each of these little round things that you're looking at are someplace that a single cell was laid down, and it grew and divided and grew and divided until you can visually see it. Otherwise, you can't see bacteria unless you have a microscope. So that's what we do. So almost the most labor-intensive part of this is subculturing them, because there are thousands of different bacteria that you can culture that are on the surfaces of sharks. So that's a pretty labor-intensive part of it.
Which part of the shark are you finding to have either the most variety of bacteria or just different amounts of and levels of bacteria?
Well, let's look. Let's see. I was going to try to wing that, but I think I have some data here, and I'm not showing all of the slides, but we're just looking at different bacteria that we isolated and genetically identified actually from the teeth, various different regions like the ampullae of Lorenzini, which is a sensory snout. And we won't, it really doesn't matter what these bacteria are. The cool thing is that we can isolate it's like over 35 genera of bacteria just that you can culture, and it's estimated that you can only culture less than 1% of bacteria in the environment. I have some fantastic collaborators, Zoe Pratt and Frank Stewart at Georgia Tech, are looking at a more comprehensive view of all the bacteria that are there, but these are just the ones that I can culture. And you can see there's quite a diversity, and if we keep running through them, sure everybody loves to see data. Yeah, there are various different regions. It almost, and I don't have them all on here, but it almost seems like the dorsal regions, just the epidermis regions, will have some of the more diverse areas as well. And the teeth have a lot of microbial diversity on them.
Was there anything that particularly surprised you as you brought these back to the lab and started seeing where and what kinds of bacteria you were finding on the sharks? Did anything jump out as like, wow, this is not what I expected, or this is exactly what I expected?
Well, a couple of things. So if we'll have to verify this by repeated sequencing, but it looks like we might have a couple of new genera of bacteria, which is kind of hard to find these days, but I'll verify that. I'll resequence those and make sure that wasn't a mistake.
What would that mean?
That means that something that's less than 83% identical to anything in the database, so it could be named a new genus.
So how would you go about? Is it just research, or like, is it a big deal to discover something new like this, and what are the implications of that?
Well, I don't know. It's not that big of a deal much anymore. It used to be a bigger deal. Now everybody has sequenced almost everything in the ocean just with mass sequencing. So I'm not sure. It'll just be most similar to, and if it's been shown to produce other secondary compounds that are of interest, or has been shown to produce something else that we're not looking for, I don't know, it could be interesting. Another thing that is kind of interesting is that when we're isolating bacteria from the blood, we tend to isolate bacteria in the blood. So the blood is not always sterile in sharks, which I think some veterinarians knew. And of course, there's not a lot of diversity, but we get kind of the same genus, some Bacillus species in there. And some of these produce antibiotics as well. So, yeah.
So, real quick, are you coordinating with like some of the vets that work with OSearch about like what you were just discussing with the blood? Like, so you're one of several collaborating scientists, right? Does that mean that you can like go to Dr. Harley Newton and start kind of collaborate with her, or have you not quite gotten to that point yet?
Well, we haven't quite gotten to that point yet, but I'm definitely already collaborating with her because she's the one that gives me the blood samples right off of the lift. So
Okay.
Yep, it's something we can look into in more detail. And I think she's noticed that same trend over the years. She's certainly looked at many more sharks than I have.
So I see that you have Hilton, who's a male white shark, and then Savannah, who's a female white shark. Have you guys looked into any other shark species to look for these antibiotic-producing bacteria?
Yeah, I think I've had Daniel Conrad's one of my students, and then Diego Gill was a postdoc in my lab, and I think maybe one of the next slides shows Daniel kind of did a survey of different shark species in Port Royal Sound in South Carolina, right off of Beaufort, and with Brian Fraser with the Department of Natural Resources. And I think in the next slide, if you can show that. Yeah, we've looked at several other species of sharks and shown that different percentages of bacteria that are isolated from those sharks produce antibiotics. So we've bull sharks, lemon sharks, black nose, black tip sharks. So there are a lot of antibiotic producing bacteria on black tip sharks. So yeah, this is kind of a study that we're in the process of writing up for publication now. Kind of interesting. And we found, I mean, I guess the best thing is that we found a lot of bacteria that are produce antibiotics against methicillin-resistant staphylococcus or MRSA, vancomycin-resistant enterococcus, and nice big zones of inhibition. And they don't appear to lyse red blood cells, and they don't appear to be small proteins. So we do other little bioassays to see if they might make potentially good drugs. So we have a lot of over 50 potential good candidates for drug discovery as well.
So what are some other studies that your students are doing in your lab or have done in your lab?
Real quick, Jen, before we get to that, because this was a good question that had to do with those slides that we did just talk about, Kim. Sorry, Jen, that this graphic is going up right over your face. I apologize. I don't know how to move it. But obviously right now we're taking samples of white sharks all up and down the East Coast right now. Are you seeing different bacteria in different places, or has it pretty much been the same bacteria that you've been finding, whether we tag the shark in Nova Scotia or we tag them down in the NASPA region?
Oh, that's such a great question. We actually do see a regional difference, and we have to get more into the data. And this is where our collaborators from Georgia Tech will be able to help a little more with that, because it's a much more comprehensive look at the bacteria that are present. But there is a local influence. But that's a great question. And the reason that's a great question is if you find the same bacteria on sharks, regardless of the location, you might hear that those are actually beneficial. So those belong on the shark. So that's kind of what I'm looking for, is that least common denominator, the bacteria that are all the same on the surface of the shark. So then I can start asking my first question, which is, are these really symbiotic with sharks? Do they belong there, and are they always there? So thanks for asking.
Okay, Jen, now you had a question.
Yeah, I was just wondering what some of the other studies were that your students are either currently doing or have done in your lab.
Well, I had a Hilton Head prep high school student come to me and ask me to do a project, and it was his idea. He wanted to look at bacteria on sharks' teeth and determine what antibiotics that we currently have available kill them. And the reason he was interested in that is for therapies for shark bite victims. So one problem if you get bitten by a shark, I mean, a big secondary problem is infection. So he's interested in what type of antibiotics he could advise doctors to use to treat shark bites with. So I think this little clip highlights his.
My name is Lucas Camita. I am from Hilton Head Island, North Carolina. I'm a student at Hilton Head Preparatory School, but I'm working with Dr. Ritchie of the University of South Carolina at Beaufort to identify bacteria found on sharks. So So when we got to the boat today, we pulled up and they already had a shark. So as soon as we got on board, we had to get the shark on the lift and jump on and take swabs and samples and everything. There were several different swabs that we had to take from the white shark that was landed, some of which included from the cloaca, the mouth, the eyes, and then what I was specifically looking for were bacteria in the mouth of the shark and trying to identify what they are and test their sensitivity to different antibiotics for shark bite research. When somebody's bitten, it's not necessarily the fact that they'll lose the limb or die, it's the risk of a bacterial infection developing. So preventing these bacterial infections by using different antibiotics and having a knowledge of what to treat the bites with. This is my high school science fair project. And when I initially started this research, I was thinking more small scale, like I'd go out, catch sharks myself. It was basically a reason for me to go shark fishing and not have my mom yell at me for spending time fishing. But it just kept growing and growing until it led to me being here, swabbing white sharks for bacteria. And I'm really excited to see where it goes in the future.
Yeah, so he ended up winning a scholarship to college with that science fair project. So any high school students out there, go hit up your local scientist to let them use your lab if you have an idea. All right, I just wanted to highlight some of the students that on this project from the University of South Carolina, Beaufort. There are a bunch of them, and one of the main ones that did an awful lot of work was Daniel Conrad. And then there's Diego Gill, who did a lot of that work as well. So.
Very cool. There are no other slides, am I right, Kim?
Yeah, that's it.
Cool.
Yeah,
So
I've
been kind of taking note on some questions here, but also want to say, yeah, that is one heck of a cool science fair project. I haven't met him, but I heard he was a lot of fun to have on the ship. So well done, and thanks for helping him get there. One question that I have here is, there's a lot of questions. Sorry, Jen, again, you got covered up. Do we completely understand white shark immune systems yet? There's been a lot of talk, do sharks get sick? Do they get cancer? How far along are we in understanding these immune systems on these animals?
That's a great question, too. So my collaborators at Moat Marine Laboratory, Carl Lure and Kathy Walsh, are experts in that. And that, from what I understand, it's not very well understood. So they have kind of an ancient immune system. It's not based on our adaptive immune system, which is based on having marrow and having white blood cells that it stemmed from that. It's based on, they don't have that. They have a cartilaginous kind of structure. So they have some other type of epiglottal, I'm going to butcher the term for that, some sort of little organ. Epiglottal that produces some sort of immune system for the sharks that Carl and Kathy are studying, and some others are studying. And they particularly, this is really interesting, they are studying this relative to whether or not this fights cancer cells, and they've actually shown that it prevents the growth of cell culture cancer cells, a wide range of them. So I believe sharks can get cancer. I think they can get sick, but I think it's just not that common. So that's a good question too.
Then the second half of this question, I think it's probably on everybody's mind, and you may not have a good answer to this, but obviously illness and sickness is in the news a lot right now for obvious reasons. If there's any chance that, what these bacteria found on sharks could have an answer to a coronavirus, COVID-19 kind of thing, or is it just?
Well, there's always that possibility. What I mentioned to everybody is that I have this pool of cultured bacteria cryopreserved that can be screened for any activity of interest, and the only thing I'm screening them for are antibiotic activities. But if you could come up with an assay for something, you can screen it for that activity, antiviral activity. Right now, I think scientists are pretty on track with the right kind of targets that they're looking for to be therapeutic for the current coronavirus. But yeah, it's possible.
So this question, have you, when you've been on the lift, have we ever tagged a shark that we found, wow, this shark looks to actually have some kind of bacterial infection that's not this beneficial bacteria that you are taking off it, that seems like this shark may be potentially sick or something?
Well, yeah, it's possible. One of the last sharks that we tagged in Nova Scotia, Vimy, had he was a pretty big shark, but he also had a couple of bite marks on him from what we got some fluid from that and some swabs from around his wound. So we're kind of looking at those bacteria. They're all frozen away, and we're waiting to assay them. But yeah, we're looking to see what types of bacteria those are in that shark. And actually, Harley Newton, the veterinarian on that particular expedition, might know a little bit more about that as well. But I know she's waiting.
So you may recognize this guy, Dr. Bob Huter. He's sort of baiting you with a question here. I think we did see a little bit of the answer to this in that video. But do you really want to anything you really want to say about driving home this point that Dr. Huter has about white shark bites and how some of this work may really help?
Yeah, but yeah, absolutely. Anything that can be introduced into a wound can be harmful and can be pathogenic and cause infections. And one thing that my student Lucas Tamita figured out is that chloramphenicol seems to kill every shark bacteria that he cultured, so across the board. So I'm not sure how much chloramphenicol is used in medical practices, but that's one that killed everything. And then we have data on other antibiotics that killed various things. So it'll depend on what the infection is caused from predominantly. But good question, Bob. Thanks.
Let me see if I can find one more here. I think we have time for one. So I think we did talk about this one a little bit, but I think it's a good way to sort of sum everything up, bring it all back. Even though we have sort of talked about this, let's just wrap it up one more time. Humans have bacteria. Obviously, answer this question. Do sharks have the same beneficial bacteria that humans have? And why it's so important that we study them, just to wrap it all up here in one bow.
Well, humans have typical bacteria in their mouth. They're not always beneficial. As a matter of fact, that's why you have to brush your teeth so you can get that biofilm off that they're constantly down. And they can actually, if you have any kind of heart condition and your dentist tells you to take an antibiotic before you come get your teeth cleaned, that's exactly why, because those bacteria, if they end up in your bloodstream, can also cause sepsis and cause infections. But if we want to go back to the slide, that very first pie slide will tell you exactly what bacteria are in white shark mouths. Might have to zoom in, and it might be hard to see, but there we go. And I don't know if you can see any of those on there. I can't see them. It's just a wide range of gram-positive, gram-negative, and marine bacteria are often different than bacteria that we find associated with us or any terrestrial organism. So that is the marine bacteria that are typically associated. And you'll see Vibrio. Vibrio is a general one, Pseudomonas type bacteria, and others. So.
Cool. So I think that we'll make that our last question. But I want to say, and I am going to turn it over here real quick to Jen, because as almost everybody, we're all working from home too. Students are learning from home. And so we really want to try and find ways to get kids engaged. And so, Jen, do you want to tell just really quick a little bit about our STEM packets, our education packets, and what we have coming up in the next couple of weeks in terms of these virtual classrooms?
Sure. So we have about 100 lesson plans available for free download, and we also have some expedition packets available as well. The NASPA expedition packet actually highlights a lot of Kim's work that she does. And we're going to be talking about that a little bit more tomorrow and go over an activity that you can do at home as well. So we'll have that happening tomorrow at 1 o'clock, and you can go ahead and register for that. The link is on our Facebook page as well as Twitter, right, John? Twitter. And then for the next few weeks, we have a lot more activities such as this, where we're going to be discussing with different scientists their work and hosting a few different discussions. And it's a wide range. We have a lot of scientists that we work with, and they all are doing amazing work for the world as a whole. So each week we will highlight a scientist, discuss their work. We'll go over some curriculum that you can use along with that to further expand your knowledge on the topic. So we have on Tuesdays, we will have an elementary school level discussion with the scientists. So yesterday we hosted that. And then on Thursdays, we're going to do a secondary, which is 7 through 12. And really, most of them you can learn on a wide range of grades. Those are just our targeted audience. So don't feel like you have to go to either one depending on your grade, where we are able to kind of approach a broad audience with them. So tune in each week. I know our plan is for about 8 weeks, so we got pretty straight through summertime that you guys can interact with us and join us. So if you have any questions, again, about the education program, go ahead and visit our website, click the education tab. You can download all the materials there, and you can always email education@osearch.org, and either myself or another one of our education ambassadors will be happy to help you on there.
Thank you all so much. Thank you for all of your wonderful questions and for being a part of this. Thank you, Dr. Ritchie. Thank you, Jen, for this awesome presentation. And we look forward to seeing all of you back here. One quick thing, if I didn't get to your question, I will scroll through them and try and answer them, or ask Dr. Ritchie if I don't know the answer, and we'll go from there. And with that, thank you, everybody.
Thanks.
Thank you.